Hello friends, today we are taking notes on dehydration synthesis and hydrolysis. The first one that we're going to talk about is dehydration synthesis. To dehydrate means to remove water, and synthesis means to build. So this is going to be building something by removing water. And because we're talking about biomolecules, we are talking about how we build a polymer from monomers by removing water. Another name for this is biosynthesis. So there are other things that can be made through dehydration synthesis, but because we're talking about biomolecules, polymers and monomers specifically, this is a type of biosynthesis, which is a type of dehydration synthesis. The way that I would write this as a chemical equation is that I'm going to take a monomer, and on that monomer, there is an OH attached to it somewhere. And then I'm going to take another monomer, and on that monomer, there's an OH, but we only care about the H from that one. So it's attached to it as well. And when I combine those by removing the OH and the H, I'm going to end up with H2O, because I've got two H's and an O. So this is going to leave the monomer, this is going to leave the monomer. Those are going to bind together to make my water. And that's going to leave this monomer needing a bond and this monomer needing a bond. And as a result, they are going to bond together. And if I do this over and over and over again, I get a polymer. Hydrolysis is the opposite. So instead of building, we are breaking. And instead of removing water, we are adding water. Hydro refers to water. Lysis means to split. So in this case, we are going to break a polymer into monomers. And we're going to do this by adding water. And this is the process that happens inside of your digestive system. And we also refer to this as digestion. So if it's the opposite, then my chemical equation is also going to be opposite. So I'm going to start with a polymer, and I'm going to add water. And as a result, this water is going to split up, and it's going to jam itself into this polymer, and I'm going to end up with a monomer that has the OH from the water added to it, and another monomer that has the H from the water added to it. So let's talk about how this works with some actual monomers and biomolecules. So I'm going to start with this one here. And this is an example of what's going to end up being a lipid. And I know that because I have here a glycerol. So I've got my 3-carbon chain, and that's telling me that this is a glycerol. And then over here, we can see that I have these carbon chains where there's a whole bunch of carbons, and the only thing bonded to them are hydrogens. And that's going to tell me that these are fatty acids. These happen to be saturated fatty acids, and I know that because there aren't any double bonds between the C's. When these bind together, we're going to do a dehydration synthesis. So we're going to take these monomers here and this monomer here, and we're going to bind them together. To do that, we need one of my monomers to lose an H, and one of them to lose an OH. And when that H and that OH are removed, you end up with a molecule of water. And that is going to be dehydration synthesis. But if I go the other way, if I start with water and I shove an OH onto this monomer and an H onto this monomer, then I can actually separate these things. And that would be an example of hydrolysis. So now I'm going to fold these in a very specific way. So I'm going to fold along this dotted line right here and this dotted line right here. And I want the letters on the outside. And what I can do now is I can fold these towards the middle, and I'm trying to get my monomers to now line up. And so I now have a C bonded to an O bonded to a C. So the way that this works is if I follow these bottom arrows where I pull these apart, pull these sides apart, then I'm going to go from my polymer and I'm going to do hydrolysis and I'm going to get a monomer and a monomer. But if I follow these arrows where I push these together, then I'm making the polymer. So in that case, I'm going to remove my H and my OH and I'm going to bind these things together to make a polymer. This is a polymer that is made up of a glycerol and 3 fatty acids. So this is an example of a triglyceride. And a triglyceride is a type of lipid. So I'm going to move and I'm going to write right here, and this is a lipid. Lipids are the polymers. They are made out of monomers. And for lipids, the specific monomers that they are made out of are glycerol and fatty acids. And this can go either way. I can either take my lipid and break it down into glycerol and fatty acids, or I can take my glycerol and fatty acids and I can build a lipid. I'm going to tape in my triglyceride by taking 2 small pieces of tape. I'm going to put 1 piece of tape on the top in the center, 1 piece of tape on the bottom in the center. I'm going to line this up where I want it to be in my notebook, and I'm going to tape it in. And now I can go back and forth. I can break these apart, hydrolysis, or I can push them together, dehydration synthesis. Okay, we're going to move on. We're going to do this 3 times total because we've got 3 different biomolecules. So the next one that we're going to be talking about is this one. And these are carbohydrates. And I know that these are carbohydrates because they have this ring shape with an oxygen inside of it. There's a ring shape with an oxygen inside of it. This one happens to be glucose. This one happens to be fructose. And you'll notice that there's an OH and an H, which means I can remove the OH and the H, making water. That would be dehydration synthesis. Or I could go the other way. I could take water and I could put an H there, and I could put an OH here, and that would be hydrolysis. Now, does it matter if I take the OH from this side and the H from this side, or could I take the OH from this side and the H from this side? It actually doesn't matter. So because there's an OH on both sides, I could lose the OH and the H in any combination as long as there is 2 total H's and 1 oxygen so that I can make water. I'm going to fold this again along the dotted lines. When I do dehydration synthesis and I bring these together, I now have 2 of these sugars, so I have a disaccharide. And you might have noticed that with these sugars, they also have an OH on this side, an OH here, and an OH down here. They have an OH here, and an OH right here. And that is going to be really important. That is really important because I can actually make long chains of these where I could take the OH from here and the H from here, and I could bind these together to make a chain, and I could do it again. I could also arrange it like this, and I could take an OH from here and an H from here. And in this way, I'm going to be able to get really long chains of sugars, and I'm going to be able to make more complex sugars where not only do I have chains going this way, but I could also have chains going this way or chains going this way, or chains off of this one, or off of this one. Either way though, these are all carbohydrates. Carbohydrate is kind of a dumb word because yes, it is the polymer, and the monomers are sugars, but I could also refer to the sugars as carbohydrates. So just know that that's a little bit weird. Now I'm also putting many sugars because sugar is one of those things that could have a whole bunch of sugars all in a long row. And so in order to be a polysaccharide, I would need 3 or more sugars. I'm going to tape this the same way that I did before. Last but not least, I've got this model here. And please notice that this one has N. N is nitrogen. And so far, only our amino acids and our proteins have nitrogen in them. The difference between these two things is this section right here. And that section is called the R group. And this amino acid happens to be an alanine. And this one is a cysteine. And the difference between cysteine and alanine is what is attached to this carbon here. Now, just like with our other monomers, I have an OH on one side and I have an H on the other side. And so just with like my other ones, I can remove those, or I can add H2O. This one is hydrolysis. This one is dehydration synthesis. We're going to do our folding. And this one is actually a little bit different. So I want you to notice that this one forms a different bond. I have a C bonded to an N. On my other molecules, I had a C bonded to an O bonded to a C. This C-end bond, the C-end bond, is called a peptide bond. The other thing that I want you to notice is that in this one, I've got an OH on one side and an H, not an OH, on the other side. So when these bond together, they're always going to bond in this order. And because there is an OH on this side, I could actually add another H on this side, and I could make a long chain of these amino acids. And between each of these amino acids, there's going to be a peptide bond. So when I actually bond many of these together, I'm going to get a polypeptide. Now, polypeptides, when they are folded correctly into a specific shape, they become a protein. So protein is the polymer, and the monomers are amino acids. The smallest proteins have about 50 of these amino acids bonded together. The largest ones have thousands of amino acids bonded together. Please tape in. And then we are not quite done. What I want you to also know about these biomolecules is that because they are biomolecules, they are organic. They are what make living things. And because they are organic, they are made of both atoms and high energy bonds. Carbon has a mass of about 12, oxygen has a mass of about 16, hydrogen has a mass of about 1. So we're going to ignore hydrogen, but I want to know how massive each of these are in comparison to how much energy they store in those high energy bonds. So to do this, I'm going to start by counting my high mass atoms. my high mass atoms are my carbons, which I am going to circle in black, and my oxygens, which I'm going to circle in red. And as I circle, I am counting how many of these high mass atoms do I have in each of these biomolecules. You should have counted that there are 23 high mass atoms. And we are going to compare that to the number of high energy bonds. Now, the high energy bonds for this molecule are a little bit harder to figure out because they're not all drawn out. So here I have a carbon bonded to a hydrogen. That's going to be a high energy bond, but there are two hydrogens, which means that there are actually two high energy bonds here. So that's 2, 3, 4, 5, 6. I'm going to keep going. Now here's another 2. And here's another 2. So in counting those, I now have 24 high energy bonds. This works out to a ratio of about 1 to 1. For every high mass atom, I have about 1 high energy bond. Let's look at my polypeptide. So we're going to do the same thing. This time though, I also have nitrogen. That's going to be a high mass, and sulfur is going to be a high mass atom. So I'm going to start with those 3 and then continue counting. Carbon is black. Oxygen is red. And in the end, for this one, I am going to have about 12 high mass atoms. Now let's look at those high energy bonds. Here, there are 3, because there are 3 hydrogens. So I've got 3, 4, 5, 6. Here there are 2. And so in total, I have 11 high energy bonds. This is still about a ratio of 1 to 1. So my proteins are going to have about 1 high energy bond for every high mass atom. Last, let's look at our triglyceride. This one has a lot more stuff. So I'm going to count in my head as I circle. All right, I counted 30 high mass atoms. Now let's count those high energy bonds. Again, there's a lot, so I'm going to count in my head. From my highlighting, you can see that this one has a whole bunch of energy. In fact, it has 64 high energy bonds. So for each high mass atom, I now have about 2 high energy bonds. And that's going to mean that for every gram of fat that I have, I store about twice as much energy as either my carbohydrate or my protein. So those are our notes. From these notes, we have learned about dehydration synthesis and hydrolysis. And we've also learned that my lipids are going to store a lot more energy in their bonds than my sugars and my proteins, even though I can stick hundreds of sugars together or hundreds of amino acids together. The ratio of atoms to bonds is still going to be about the same. So in terms of energy storage, lipids, they're the way to go.